Chapter 9. The Digestive System. The Pancreas. The pancreas is a soft, oblong gland about 6 inches long and 1 inch thick. It is found beneath the great curvature of the stomach and is connected by a duct to the duodenum of the small intestine. The pancreas is divided into a head, the part closest to the duodenum, the body, the main part, and the tail. Internally, the pancreas is made up of clusters of glandular epithelial cells. One group of these clusters, the islets of Langerhans, or the pancreatic islets, form the endocrine portions of the gland and are therefore part of the endocrine system. Some of these clusters consist of alpha cells that secrete the hormone glucagon. Other clusters consist of beta cells that secrete the hormone insulin. The other masses of cells are called the acinite, which are the exocrine glands of the organ. The acinite release the mixture of digestive enzymes, lipases, carbohydrates, and proteases called the pancreatic juice, which leaves the pancreas through a large main tube called the pancreatic duct, or duct of Wiersung. The duct cells secrete sodium bicarbonate. In most individuals, the pancreatic duct unites with the common bile duct of the liver and enters the duodenum in a common duct, originally called the ampulla of Fata but now is called the hepatopancreatic ampulla. The functions of the pancreas are, therefore, twofold. The acinite secrete enzymes that continue the digestion of food in the small intestine, and the alpha and beta cells secrete the hormones glucagon and insulin that regulate and control blood sugar levels. As you can see, this is the anatomy of pancreas. The liver. The liver is one of the largest organs of the digestive system. The liver weighs approximately 4 pounds and is divided into 2 principal lobes, the right lobe and the left lobe, each separated from one another by the falciform ligament. The lobes of the liver are made up of numerous functional units called globules. The functions of the liver are so numerous and important that we cannot survive without it. The liver has 6 major functions. 1. The liver manufactures the anticoagulant heparin and most of the other plasma proteins, such as prothrombin and thrombin, that are involved in the blood clotting mechanism. 2. Kupffer cells of the liver phagocytose certain bacteria and old, worn-out white and red blood cells. 3. Liver cells contain various enzymes that either break down poisons or transform them into less harmful substances. If the body cannot break down and excrete certain poisons, it stores those poisons. When we digest proteins into amino acids, the amino acids go to the mitochondria to be converted into ATP. This process produces ammonia as a waste product, which is toxic to cells. The liver cells convert ammonia to urea, harmless, that is then excreted by the kidneys or the sweat glands. 4. Excessive newly absorbed nutrients are collected in the liver. Excess glucose and other monosaccharides can be stored as glycogen, animal starch, or converted to fat. When needed, the liver can then transform glycogen and fat into glucose. 5. The liver stores glycogen, copper, and iron, as well as vitamins A, D, E, and K6. The liver produces bile salts that break down fats. These bile salts are sent to the duodenum of the small intestine for the emulsification, breakup, and absorption of fats. As you can see, this is the anatomy of liver. The gallbladder. The gallbladder is a pear-shaped sac about 3 to 4 inches long located in a depression of the surface of the liver. Its lining, like the stomach, has root eyes that allow it to expand and fill with stored bile. The gallbladder's function is to store and concentrate the bile produced by the liver lobules until it is needed in the small intestine. The bile enters the duodenum through the common bile duct. The small intestine, the major portion of absorption and digestion, occurs in the small intestine. It is approximately 21 feet in length and averages 1 inch in diameter. The small intestine is divided into 3 portions. First is the duodenum, which is the shortest part, and is about 10 inches long. The duodenum originates at the pyloric sphincter and joins the second portion, the jejunum. The jejunum is about 8 feet long and extends to the third part, the ileum, which measures 12 feet and joins the large intestine at the ileocecal valve. The mucosa of the small intestine contains many pits lined with glandular epithelium. These pits are known as the intestinal glands or crypts of Leverkusen. They secrete the intestinal digestive enzymes that supplement the bulk of the digestive enzymes secreted by the liver and the pancreas. The submucosa of the duodenum contains numerous Brunner's glands, now called duodenal glands, which secrete an alkaline mucus. Additional mucus is secreted by goblet cells. This mucus protects the walls of the small intestine from being digested by enzymes and neutralizes the acid found in the chyme. Chyme is the term used to describe the digested, viscous, semifluid contents of the intestine. Approximately 80% of all absorption of nutrients, simple sugars, amino acids, fatty acids, water, vitamins, and minerals occurs in the small intestine. The anatomic structure of the small intestine is highly specialized for this function. First of all, the tract is 21 feet long. Second, an even larger surface for absorption of nutrients is provided by the structure of the walls of the tract, which are thrown into a series of folds called plicae. Third, the mucosal coat is transformed into projections called villi, which look like microscopic eyedropper bulbs approximately 0.5 to 1 millimeter long. A tremendous number of villi line the intestine, about 4 to 5 million. These villi vastly increase the surface area of the epithelium for absorption of nutrients. The structure of each villus contains a capillary network where blood picks up nutrients, a venule or small vein to transport the nutrients, an arteriole or small artery, and a lacteal of a lymphatic system to pick up fats. In addition, the individual epithelial cells that cover the surface of a villus have a brush border of microvilli to further increase the absorptive capability of the small intestine. Nutrients that pass through the epithelial cells covering the villus are able to pass through the endothelial cells of the capillary walls and through the lacteals to enter the blood and lymphatic circulatory systems. From there they are transported to the billions of cells in the body. As you can see, this is the structure of the small intestine. And this is the absorptive features of the small intestine. A. Plicae circulars. B. Villi. C. Microscopic anatomy of a villus. D. Microvilli. The large intestine. The functions of the large intestine are the absorption of water, the manufacturing and absorption of certain vitamins, and the formation and expulsion of the feces. The large intestine is about 5 feet in length and averages 2.5 inches in diameter. It is also referred to as the bowel. It is attached to the posterior wall of the abdomen by extensions of its visceral peritoneum known as the mesocolon. It is divided into 4 principal regions. 1. The cecum, the pouch-like first part of the large intestine. 2. The colon, the largest part. 3. The rectum, and for the anal canal, the opening from the ileum of the small intestine into the cecum of the large intestine is a fold of mucous membrane known as the ileocecal valve. This valve allows material to pass from the small intestine into the large intestine. The cecum, the blind pouch one end is closed, is 2 to 3 inches long and hangs below the ileocecal valve. Attached to the closed end of the cecum is the twisted tube known as the vermiform vermi-H-form appendix, about 3 inches in length. The open end of the cecum merges with the long tube called the colon. The colon looks like the tube of consecutive pouches. The pouches are called haustri. The first part of the colon is known as the ascending colon. It rises on the right side of the abdomen, reaches the under surface of the liver, and turns to the left at the right colic hepatic flexure. The right colic flexure continues across the abdomen to the left side as the transverse colon. It then curves beneath the lower end of the spleen to the left side as the left colic splenic flexure. Next it passes downward as the descending colon. The last part of the colon is called the sigmoid colon, where the colon joins the rectum. 3 mechanical movements occur in the large intestine. Haustral churning peristalsis at the rate of 3 to 12 contractions per minute. Mass peristalsis. Food in the stomach initiates mass peristalsis, which is a strong peristaltic wave that begins in the middle of the transverse colon and drives its contents into the rectum. The rectum is the last 7 to 8 inches of the gastrointestinal tract. It is situated anterior to the sacrum and the coccyx. The terminal 1 inch of the rectum is called the anal canal. The mucous membrane of the anal canal is arranged in a series of longitudinal folds called the anal columns that contain a network of arteries and veins. The opening of the anal canal to the exterior is called the anus. It is guarded by an internal sphincter of smooth muscle and an external sphincter of skeletal muscle. The absorption of water is an important function of the large intestine. In addition, bacteria in the colon manufacture 3 important vitamins that are also absorbed in the colon: vitamin K needed for clothing, biotin needed for glucose metabolism, and vitamin B5 needed to make certain hormones and neurotransmitters. Mucus is also produced by glands in the intestine. Intestinal water absorption is greatest in the cecum and ascending colon. As you can see, this is the anatomy of large intestine.